So welcome everyone to our EASD Journal Club. I'm very happy that a couple of people showed up. I hope there's more people coming once we started. And today it's my honor to introduce Estela Lorza-Gil, and she's currently a senior scientist at the Island Lab in the Institute of Diabetes Research and Metabolic Diseases of the Helmholtz Zentrum Munich at the Eberhard Karls University Tübingen. So that's quite a long name. Short version is IDM. So you probably know it like that. So we're the guys in Tübingen doing diabetes research. She started her education with a Bachelor in Biological Science in Spain, León, and then moved to Brazil to Campinas for her PhD. And she told me she's been there for 6 years, but then she felt like she had to move on, which is good for us because now she's here. She also did a postdoctoral position at the Pharmacology and Toxicology in Bonn before she joined us at the IDM. And I'm very happy to hear about her paper in Diabetologia today. And I'm also congratulating you because you just got a DZD twinning grant for the follow-up for this study that you will present today. So Estela, the stage is yours. Thank you.
So thank you very much for the nice introduction, Simon, and also thank you for inviting me to be a speaker for this journal club for the EASD. I will just share my presentation. You can see it, isn't it?
Yes.
So, so as I said, I am really excited to present here this paper. So it was published in Diabetologia last year, but also I'm even more happier because this paper is not finished. So it opened like a lot of questions, and we had already a follow-up project. So I wish I have some nice feedback out of it from all of you afterwards. So yes, the paper is entitled Glucose-Stimulated Insulin Secretion Depends on FFA1 and Gq in Neonatal Mouse Islets. So when I first joined the lab of Susanne Ulrichs during my first postdoc, the lab was interested in studying the mechanisms of beta cell failure in obesity-related type 2 diabetes. And we knew that the best functional characteristic of mature beta cells is the glucose-stimulated insulin secretion. And this characteristic is it develops gradually during the postnatal state of the life. And in utero programming, but also this neonatal environment characterized by lipid-rich diet from the maternal milk, but the carbohydrate-rich diet after weaning can influence the outcome of these mature beta cells at the end of the functional maturation. And it is known that obesogenic maternal diet can negatively influence in the functional maturation of the beta cells, with an increase in the incidence of type 2 diabetes in the offspring. But also in the adult life, obesity is a well-known risk factor for development of type 2 diabetes, promoting dysfunction of beta cell failure, adapting neonatal-like phenotype, a reduction in the beta cell mass, and impair glucose-stimulated insulin secretion. And the high levels of non-esterified fatty acids produced by obesity are recognized stressors that impact on the beta cell function, but also can stimulate the insulin secretion in the beta cells by the activation of the FFA1 receptor. And this receptor, the free fatty acids receptor, is a GPCR that is activated by long-chain fatty acids. It's highly expressed in beta cells, and it's known, so as I said, so mediates the stimulatory effect of free fatty acids and on insulin secretion. But although the long-chain fatty acids are the major compound of the maternal milk, how or whether FFA1 receptor can modulate the functional maturation of the beta cells is not known. So that's why we ask whether FFA1 receptor is involved in the neonatal beta cell function. If FFA1 in parental high-fat diet, so if FFA1 is involved in parental high-fat diet, induce dysfunction of progeny beta cells. So in order to answer that questions, we use wild type and FFA1 knockout mice. So these mice express GFP proteins in beta cells and red fluorescent protein in the other cells of the body. And these animals were exposed to chow diet and high-fat diet for 8 weeks before the breeding, but also during the pregnancy and during the lactation. And we analyzed the offspring, the F1 offspring, and at the different postnatal days, and also after the weaning, where the animals were exposed to chow diet. So we analyzed the offspring at the P1, so P states for postnatal day, so P1, 1 day old, P6, P11, and P26. So we had then 4 groups. So the offspring of a wild type chow diet, so wild type offspring parental chow diet fed animals, wild type high-fat diet, FFA1 knockout chow diet, and FFA1 knockout high-fat diet. From all these groups, we analyzed in the offspring the pancreas, the beta cell proliferation after isolation of the pancreas, and also the beta cell mass. And we isolate the pancreatic islets, and we perform insulin secretion and transcriptome analysis. So what we found was, like, in the P6 offspring, so in the 6 days old neonates, the high-fat diet feeding, but also the FFA1 deletion increase the blood glucose levels, which suggest like defects on insulin secretion or impair insulin secretion. Therefore, we isolated the islets of these mice and performed glucose-stimulated insulin secretion assay. And as you can see here, so in the wild type offspring, the high-fat diet didn't affect the insulin secretion. It was very similar between these groups. However, in the FFA1 knockout offspring, even in the chow diet fed animals, the glucose-stimulated insulin secretion was quite low. And in the knockout and high-fat diet animals, they didn't respond to glucose. So not only the FFA1 knockout had an impairment in the glucose-stimulated insulin secretion, but also when the islets were stimulated with palmitate. So as is expected, when in the FFA1 knockout, islets don't produce insulin secretion in response to palmitate. However, when the islets were stimulated with forskolin, so forskolin can activate adenylyl cyclase directly without bypassing the activation of the receptor. So forskolin could stimulate insulin secretion in all groups in a similar way. But when we analyze the effects in the P11 offspring, so 11 days old mice, the negative effects of the deletion of the FFA1 disappear in the blood glucose, but also in the insulin secretion. So we can see here that in the knockout chow diet, the glucose-stimulated insulin secretion was very similar in comparison with the wild type. We only observe a reduction in the insulin secretion in the FFA1 knockout with parental high-fat diet feeding, but only after in the incubation of 20 millimolar glucose. And as we observe in the P6, we can see here that palmitate wasn't able to induce insulin secretion in the knockout progeny, independently of the diet, and forskolin induced the insulin secretion in a similar way among all groups. So with all this data, what we got was, so this data suggests that FFA1 signaling can promote neonatal glucose-stimulated insulin secretion, but also this receptor is necessary for the adaptive insulin secretion through parental high-fat diet. And we were very surprised to observe that indeed FFA1 is responsible for the glucose-stimulated insulin secretion. But this is it was really new, but only was at the P6, so at the six days old neonatal islets. That's why we focus our research in analyzing the effects of the FFA1 in beta cell function at this age, so at 6 days old. And in order to understand this impaired glucose-stimulated insulin secretion after the deletion of the receptor, we thought that maybe the beta cell maturation could be impaired, and we therefore performed like a transcriptome analysis. So we did like an RNA-seq comparing the neonatal islets from the wild type with parental child diet feeding versus the FFA1 knockout offspring with parental child diet feeding. And so when we got the data from this RNA-seq, we got a bit disappointed because no beta cell markers were altered in the FFA1 knockout, and we found only 24 genes that were upregulated in the knockout in comparison with the wild type. And the top ranking genes of these 24 genes, we can I show all of them here in this figure. So the top ranking are genes related to proliferation, and most of the genes that were upregulated, and I marked here with this black triangle, are genes associated with a juvenile beta cell phenotype. That's why we wanted to analyze the proliferation of these animals during the postnatal life, from the P1 to P26. And what we saw was that in the FFA1 knockout offspring, indeed we observed like a peak of proliferation at P6, so like at early stage of the differentiation, but this increase in proliferation wasn't sustained as in the young adult mice. The proliferation levels were very similar to the wild type. And in the offspring from parental high-fat diet, the proliferation was increased in comparison with the child diet, but the peak of proliferation was in later stage of the development of the beta cell, so at day 11. Despite of having this increase in proliferation in the FFA1 knockout, we didn't see any change in beta cell mass, and also we didn't see any change in pancreatic insulin content. So with what all this data suggests was that in the FFA1 knockout progeny from parental child diet feeding, in neonates, there is an increase in the beta cell proliferation, but a juvenile genotype, so immature phenotype. So as I mentioned before, so FFA1 receptor is a GPCR that potentiates glucose-stimulated insulin secretion by recruiting GQ protein. And to see if this is also the case for our neonatal islets, we treated the islets with a GQ inhibitor. This inhibitor was kindly provided by Effie Constantins from the University of Bonn. And we tested the inhibitor in the wild type P6 offspring. And we measured then insulin secretion. And as you can see here, we saw like a nice glucose-stimulated insulin secretion. Then palmitate could potentiate insulin secretion, but also TUC469, which is a FFA1 agonist, could potentiate this insulin secretion. But in the presence of the inhibitor, of the GQ inhibitor, not only palmitate and TUC stimulated insulin secretion was blocked, but also glucose-stimulated insulin secretion was completely gone in the presence of the GQ inhibitor. And to confirm this experiment, we also treated the cells with another GQ inhibitor, and we saw very similar results. So forskolin is known to activate adenylyl cyclase and in beta cells stimulate insulin secretion, bypassing the activation of the receptor. And this we can see here in the FFA1 knockout islets, where forskolin induces the insulin secretion. But interestingly, what we found was that when we treated these islets with the GQ inhibitor together with forskolin, we saw a reduction in the forskolin-induced stimulated insulin secretion. But it's something that we couldn't explain, so we don't understand how GQ signaling, so the inhibition of the GQ signaling can interfere in the DS signaling. So we did, in order to clarify that for sure, we need to perform more experiments. But what we thing that is happening is that adenylyl cyclase is somehow regulated positively by PKC and calcium, which are downstream targets of GQ signaling, as it has been already shown in pancreatic islets from several authors. We then hypothesized that the GQ inhibition could, in pancreatic islets, could then repress some inhibitory proteins, like for example DIO proteins, and in that way would reduce the insulin secretion that we can see with the inhibitors. And that's why GQ could interfere in the GI proteins. So to test this hypothesis, we treated we use pertussis toxin, which is a drug commonly used to block the DIO protein. And we use pertussis toxin together with the GQ inhibitor. For a short period of time. So pertussis toxin was treated for 18 hours and then only incubation of 2 hours with the GQ inhibitor before measuring insulin secretion. So we tested this hypothesis in the wild type P6 offspring, and we measure insulin secretion. And as you can see here, so we have a nice glucose-stimulated insulin secretion, which is blocked by the GQ inhibitor. But the presence of pertussis toxin, where the DIO proteins are blocked, we can see a nice induction of insulin secretion by pertussis toxin. But when pertussis toxin is together with the GQ inhibitor, there is some reduction in this pertussis toxin-induced insulin secretion. And this was also confirmed or tested using FFA1 knockout. Neonatal islets, we can see here that there is no glucose-stimulated insulin secretion due to the lack of the FFA1 receptor, but then in the presence of the pertussis toxin, the blocking of the DIO induced an increase in insulin secretion, but in the presence of the GQ inhibitor, this induction is completely gone. So which suggests that the pertussis toxin glucose-stimulated insulin secretion is sensitive to the GQ inhibition. But FFA1 receptor is not the only receptor that can potentiate insulin secretion in beta cells. We have also GLP-1 receptor that stimulate insulin secretion, mainly recruiting the DS protein. But a couple of years ago was published a paper where they show that some islets can stimulate glucose, so that some islets glucose-stimulated insulin secretion by GLP-1 receptor is mediated by GQ signaling, but especially in those islets that are under metabolic stress conditions. So to test this hypothesis in our neonatal islets, we incubated the islets with the GQ inhibitor and stimulated with exendin-4, which is a ligand of GLP-1 receptor in order to stimulate the pathway. And what we can see here, so we tested in the neonatal islets, wild types, and you can see a nice stimulation of glucose stimulated insulin secretion, but then is potentiated by XNF4. And in the presence of the GQ inhibitor, is partially blocked. It's partially blocked the induction of glucose-stimulated insulin secretion. So here is the last slide with the results. So because we wanted to know if the dependency of glucose-stimulated insulin secretion on GQ was restricted to neonatal islets. And to answer that, we use adult mouse islets, and we treat them with the GQ inhibitor. And indeed, we observe that GQ inhibitor in adult islets, the glucose-stimulated insulin secretion is also impaired. And in these adult islets, we analyze the influx of calcium after high glucose levels, and we can see in the presence of GQ inhibitor. And as you can see here, there is a rather small reduction of this rise of this peak of calcium after high glucose levels in comparison with the control ones. And from our opinion, we don't think that this reduction in the peak of calcium is the responsible of the huge impaired insulin secretion that we can see in the human islets. But when the islets are exposed to depolarized agents like KCl, we can see that there is a peak in the influx of calcium, and there is no difference anymore in the GQ inhibitor or in the control. So it's the influx of calcium intercellular calcium is the same after depolarizing the membrane. So to see if this influx of calcium will produce the release of insulin secretion, we treated the islets with KCl to depolarize the membrane, and we measure insulin after treatment with GQ inhibitors. And as we saw in the calcium experiments, we can see that there is no alteration in insulin secretion after KCl treatment together upon GQ inhibition. And we observe similar results in human islets. So the GQ inhibition disturbed the glucose-stimulated insulin secretion, however, didn't affect the KCl exposure to the islet, since the insulin secretion was similar than in the control islets. So putting together all this data to sum up, I would say that FFA1 promotes the gain of glucose-stimulated insulin secretion in neonatal islets. FFA1 is necessary for adaptive insulin secretion to metabolic challenge like parental high-fat diet. FFA1 promotes functional beta cell maturation, and neonatal glucose-stimulated insulin secretion depends on active GQ, which is the main protein involved in FFA1 signaling. So what we think about, so when we wanted to put together all these findings, we conclude that FFA1 and GQ mediate glucose responsiveness of neonatal islets. And this is so because in neonatal islets are forced to cope with this lipid-rich maternal milk, which engage the FFA1 receptor and alpha-Q signaling in order to effectively induce the insulin secretion in response to glucose. And alpha-Q signaling can also negatively regulate the alpha-IO proteins in order to also potentiate the insulin secretion in response to glucose. And also alpha-Q can mediate GLP-1 receptor signaling. And this FFA1 and alpha-Q activation is even more important when there is some kind of obesogenic maternal environment where the long-chain fatty acids are increased and GLP-1 receptor alpha S signaling is impaired. So with that, I would like to thank to all co-authors of this paper, especially Felicia Gerst and Susanne Ulrich, and also to the people of the islet lab, and thank all of you for your attention.
Thank you, Estela. Thank you very much for this very interesting talk and the nice data you shared. So for anyone who's joined over time, we're a nice intimate round, so you can unmute yourself and ask questions if you like, or if you have any burning questions. You can also raise your hand and I will moderate. And if you're a little bit shy or you don't want to talk because you, like me, have a virus and your voice is cracking, maybe just write your questions in the chat and then I can read them out. So of course, I have some questions prepared and gotten over your talk, so I might as well start until you other people might also have some questions ready. So the FFA1 is the free fatty acid receptor, right? So you always show the glucose stimulated insulin secretion. So is it known how glucose interacts with this receptor, how it stimulates it, and if not, how would you say that happens?
So it's indeed it's not known. That's why we are really interested in finding the answer. So glucose can activate DQ signaling in a different way. So either glucose and glucose metabolism activate the GQ directly, the GQ signaling, or glucose induce insulin release, but also release of other mediators that can, in an autocrine way, bind to the FFA1 or to the GPCR coupled to the GQ signaling. In that way, could have glucose, so glucose activate the GQ pathway. And it's known that beta cells can release these other mediators together with insulin, like serotonin or nucleotides. But there is a recent paper, like in 2018, by Stefan Offermans, where he showed that the glucose stimulate insulin secretion also stimulate the release of 20-hete. 20-hete is a metabolite of arachidonic acid released by the beta cells, and it binds to the FFA1 receptor. So this would be like the easiest answer, but of course we need to test it to see if, first of all, if neonatal islets release 20-hete after stimulation with glucose or not, or
so just throwing that on there is probably the next valid step to test this. All right, any questions from the audience so far?
Yes, so thank you, Estela, for a wonderful presentation and of course the interesting findings. So I have a general question. When you are presenting, I was curious. Well, I come from East Africa, Tanzania, and what we're experiencing is that most of the children, I'll speak in a clinical aspect, and I say malnourished. So they're not really well fed, and weaning is done very early. So I just wanted to ask, what do you think of the impact of this when it comes to the prevalence of diabetes, which is projected to increase maybe in the 20 coming years in sub-Saharan Africa? Could this have anything to do with the beta cell function?
I think so it's known that weaning, so the step from breastfeeding to weaning is really important for the beta cell maturation. This is was shown some years ago, since they made some experiments with mice where they kept the animals after weaning with like a very similar diet to the maternal milk. And in those animals, the beta cell maturation was really impaired or not so good as in the animals that were weaning to the carbohydrate diet. So of course, I think the weaning to the carbohydrate diet is really important for the beta cell maturation. I don't know if I answer correctly your question.
Well, yes. And if I can ask a follow-up question, it means that, for instance, if early weaning was done, in this case, in your experiment, it will most likely, if at all it was a high fatty diet or a high carbohydrates diet after weaning, it would have a different effect either ways, right?
Yes.
Yeah, thank you. And for the free fatty acids, of course, that would lead to high insulin production in comparison to high carb diet. Is that correct?
Yes, we observe that.
Yeah. Okay, very interesting. Thank you.
Okay, thank you. Any other questions from the audience?
If I may, I had kind of a follow-up question about what you said about possibly autocrine effects. Here you looked at whole islets, isolated islets. I don't know if FFA1 is also expressed in alpha cells or delta cells. And have you thought about any ways to distinguish effects on alpha cells or on beta cells?
Yes, this is something that we really wanted to focus on, because we all our experiments were with isolated islets, like intact islets, and we talk about general action of the GQ inhibitors. And when I say like I focus in beta cells, of course GQ can inhibit receptors in alpha cells and reduce the glucagon secretion. So it's something that we know, so we are aware. We need to go deeper in studying the other endocrine cells. But we couldn't do it because of the limitation of the study, because we work with neonatal islets, and we cannot isolate as much as islets than in our adult mice. So this is something that we will do it in the follow-up project, since in the grant that we got the grant for the collaboration with Elisabeth Kempter from the LMU in Munich, and we will use pig islets, and we will get like a lot of islets, and we will induce differentiation in vitro, and we hope that we can not focus only in insulin, but also in glucagon and somatostatin. Because I didn't mention, because so when we performed the transcriptome analysis, we got 8 genes that were downregulated in comparison with knockout, downregulated in the knockout when compared with the wild type. So only 8 genes. One of them was FFA1, of course, but another one was somatostatin. So, and we confirmed this data doing PCR. So somatostatin was low, but this wouldn't explain our results, because I mean, if we have lower levels of somatostatin, we will have the other way around effect. So it doesn't explain why we have this impaired insulin secretion. So still, we of course need to go deep and try to find that.
Yeah, maybe I can follow up on that, because with your P11 islets, you showed that you have more or less the same blood glucose, but you have less insulin secretion at that point, because it's still down with the knockout, at least with a high fat diet, but the blood glucose is the same. So how do they compensate? Where does it go? Are they moving more, because then the glucose would be taken up into the muscle without insulin?
So in the P11, so I showed P11, but it's the same for P26. And in the P11, the glucose levels are the same, yes, and the insulin secretion is very similar. I don't know what you mean. So it's very similar, only in
with the high fat.
at 20 millimolar glucose, at very high non-physiological levels of glucose. But
Okay.
at 20 millimolar is similar.
Okay. Yeah. So maybe I
But we
got that. Yeah.
didn't see insulin sensitivity, so we also cannot answer. We just focus on the beta cell function. That can
Yeah. Did you observe difference in movement? So, or did you not look at that?
No, I didn't. I think if yes, I didn't realize that. Yeah, we didn't. We had many groups. Yeah.
Yeah. And you showed that there's elevated proliferation, but not really more mass. So where do the cells go?
Question.
So are they dying? Are they misdifferentiating? Or
yeah, we also ask that to ourselves because we really reproduce the proliferation data in different approaches. So there is more proliferation in the knockout in the neonatal islets, so at P6. Where is going this proliferative beta cells? We don't know, but we know that proliferation in neonatal, in the early stage of the beta cell maturation, doesn't imply maturation. So it can be that this increase in beta cell proliferation is produce immature beta cells that can now go further to be mature beta cells that can be dedifferentiated. And this is explained by other authors where they really analyze beta cell proliferation in fetus and adult, in fetus islets and neonatal and adults, and they show that this like a high increase in proliferation during the early stage of the life doesn't imply like a better beta cell mass.
Okay, thank you. Any other questions from the audience before I keep spamming? Yes, please. Lucy, go ahead.
Yes, Estela, I'm just thinking out loud again. If for instance, in your experiment, you tried to switch the diet, did you check any differences, or did you try to hypothesize maybe you could reverse some of the transcriptomic changes that you saw in the beta cells?
What do you mean, switching the diet?
Yes, for instance, if we altered the free fatty acids and we switch them back, maybe in a certain period of time, could that have a difference, or does that leave a permanent mark on it?
So this is a good point. I think to answer that this could be done maybe in vitro, having cells that can be, because there are some models where you can get immature beta cells and can be differentiated in vitro. And maybe you can play with different substances or different like lipids and glucose concentration or protein, and then we can see what affects the beta cell function at the end of or not. For example, this can be done with neonatal peak islets, because neonatal peak islets they are immature, completely immature, but there are some protocols where you can differentiate them into mature beta cells. So maybe it's something that, thank you, I will think about it. Yeah.
Okay, thank you. So are there any more questions? Because otherwise I just wonder, because I don't know, are there any clinical trials or ideas to feed basically young children with a FFA1 agonist to make sure that they do not have any negative aspects from obese parents? Sorry.
Mm-hmm. Yeah, no, I don't think it can be viable at the moment. Yeah, no.
Too early.
Because GQ signaling is not only produced by FFA1 in beta cells, so it's what we believe is that in neonates, FFA1 so GQ is really linked to the FFA1, which is not the case in adults, because mouse adult islets from FFA1 knockout, they have like a nice glucose stimulated insulin secretion. It's only in neonates where there is this, so they cannot cope to release insulin secretion by a glucose stimulation. So it's something that maybe this GQ signaling is engaged in neonate islets. Why? Yeah, it's difficult to.
Okay, so maybe out of interest, probably this is also due to the sampling problem will be addressed with the pigs, I hope. So you showed the calcium for the adult islets, but not for the neonatal islets. Do you know if there's a difference in the cytoplasmic calcium response or the insulin content with the neonatal islets?
Yeah, this is something that we wanted to do as well, because we know that it can be some kind of changes in the influx of calcium in the FFA1 knockout neonates in comparison with the wild type. We could do it only in adult islets because of limitation problems. We didn't do it for this paper, but we plan to answer that in the follow-up project. Yeah, this is something that's planned in this. That's the grant where we will work with transgenic pigs, and that they are these islets from these pigs. They express like a calcium sensor, and hopefully we will be more focused on these details. Yeah.
Okay, that sounds super cool, and I'm looking forward to hearing about these projects. So, are there any more questions from the audience?
Maybe if I may, Simon, I had a question about when you said for treatment, whether use an agonist or antagonist of FFR1. I'm not really sure, based on what you've shown and what is known in the literature, because I suppose in obese people, you will overactivate FFR1 because you will have more free fatty acids. But with your data, you show that depletion of FFR1, so inhibition of activation with free fatty acid, is kind of detrimental for beta cell maturation. So in your opinion, what is the best thing to do? Do you think we should try to overactivate or to inhibit FFR1 signaling to improve beta cell differentiation?
You will overstimulate something that is already stimulated, because in obese you have like high levels of fatty acids. So I'm not sure if like giving more drug, like it will impact on the function of the receptor. Do you know what I mean?
Yeah, I think if you overstimulate, but I was thinking if you kind of try to block, because kind of blocking the effect of obesity will be trying to block the free fatty acid receptor. But I don't know if it's a strategy. It's a very naive and blue question.
It will impair glucose-stimulated insulin secretion, so it will really be not beneficial for type 2 diabetic patients.
Yeah.
And do you think, for example, on the opposite side, people who are underweight, like people suffering from anorexia and who gives birth, they will have very low levels of free fatty acid? Do you think it can impair pancreatic development?
It's so it was some FFA1 agonists that were given to patients like non-obese, and the patients got some toxic effects in the liver, and that's why FFA1 at the beginning was like a nice therapy against type 2 diabetes and obesity, but it was suddenly stopped from the clinical studies. Why FFA1 was toxic in the liver, I don't remember right now. I cannot say it.
Okay, that's interesting. Thank you.
Maybe one last thing, since we're the EASD Academy and we're also very interested in career development and how other people go about their careers and their steps to progress in their careers, I was wondering maybe you could share a anecdote or something, one career defining moment during either this project or your journey in general, or maybe some realization that you had that led you to this point and to choose a career in academia.
I think my personal career development was divided more in countries rather than in steps, because as we were talking before, so I studied biology in Spain, but I didn't have any idea about doing research. So I just learned about biology, but how I could work in a lab, but I didn't put the hands on it. But it was only when I moved to Brazil where I started to learn how to do research, and I really like it, and I was really interested. And at that moment, I could see myself in the future as being a scientist, working in a lab with different projects and developing new ideas and so. And I think in that moment was good because I did my PhD so I really liked the study of metabolic disease, and in that moment I got my PhD in molecular biology, but I got experience in pancreatic islets. So I learned how to work with pancreatic islets, which cannot be. It's not easy, the handling and everything. So I think that's why I got my position so I got my first postdoctoral position in Germany. So I wanted to go back to Europe to be close to my family because I'm from Spain, and I moved so I did my postdoc in Germany, but I did my postdoc, but for me it was like a second PhD because I did my PhD in Brazil with another culture, another way of doing research. People were different. Language was different. And then I moved to Germany, to Tübingen, which is a really, really nice place. And I was lucky I had a really good mentor, so Susanne Ulrich, so it was really, really nice. But it was some kind of challenging. But it's something that I don't regret to going abroad while you are doing science, because I think we need to know how to work, how to do science in different environments. So because sometimes you have like a nice environment, sometimes you don't have a so nice environment. You need to face problems that you would never face in another country. Also people, the character of people, you need to learn how to deal with that person. So I think, in my opinion, career development, not only going up step by step in research, like student, PhD, postdoc, whatever, you really need to go like in a horizontally way and seeing like more the personal career development. So trying to find other cultures, other methodologies, and other people. Yeah.
Estela, thank you very much for sharing your data, your career, and answering all of our questions. So I'm very happy that we have this great journal club. And just some general announcements. So our next seminar or webinar is scheduled for mid of February, and it will be a webinar on writing ERC grants by the ERC Commission. And in March, we will have, at beginning of March, we will have our next journal club about the direct study. So stay tuned. Make sure you read the newsletter and sign up for these great activities, and also follow us on our social media. And if there's no more other remarks, I'm happy to have this nice night, and I'm wishing you an even better night from now on, and hope to see you back soon.
Thank you, Simon. And thank you all. Bye-bye.
Thank you.